Tonight, I'm really excited to present to you tonight. My name is MacKenzie Howard. I've been a member of the faculty of the Department for Neuroscience for just about a year and a half now, so I'm still a newcomer to Austin. I'm really excited to be joining this department and getting a chance to talk to you about the things that I care a lot about. So welcome to UT Brainstorms. This is a conversation, or a series of conversations about the brain and about the types of research we do and the types of topics that we're interested in the Department of Neuroscience. So I want to point out to you before I start, this is me. And tonight we have some really special guests to serve on the panel with me for the Q&A session afterwards. Dr. Lee and Dr. Keogh. Dr. Lee is a member of the Department of Psychology and also a member of the Institute for Neuroscience, which is the neuroscience graduate program here at UT. And Dr. Keogh is a member of the Dell Med School Departments of Pediatrics and Neurology, and so someone who knows a lot more about human epilepsy than I do. She is also the director for the Child Neurology Residency Program that has been established for a few years and is, or for a while now, and is now growing with Dell Med School coming and expanding here on campus. So I'm really excited to sit down after the talk, which will be maybe 45 minutes or so, and then we'll have a little break, and then we'll sit down and answer any questions you might have. So on that note, you're welcome to write your questions down as we go on the note cards around, and there's plenty of them if you come up with more than one question. I think that we also will have a microphone that we can pass around during the Q&A period, so if you have more questions that come to mind during the Q&A, you'll get a chance to ask those as well. And I want to point out again, also before we start, that the next part of this series is going to be presented by Dr. Mike Mock, who is our professor and chair of the Department of Neurosciences, so the person that I have to keep happy. And he's going to be talking about the broken brain, a conversation about how brain damage informs us on brain function. And one more note about the UT Brainstorms program is we recently have kind of made a commitment to continue this through next year, as far as I know, and so we're going to have several dates. So look for announcements starting, I'm not sure if it will start in August or September, but I think it will start up again. Of course, at Dr. Mock's presentation, he'll have more info on that. And we also have some cool t-shirts that you'll notice some people wearing tonight. And those will be available, and there will be more details on getting those at the next part of this series for sure. So with that, I'd like to get started and tell you a bit, or start this conversation about neural signaling and epilepsy and the electric brain. And something that I'm really interested in is the brain and how the brain uses electricity. So the brain does in fact use electricity. And this, when I first learned about this as an undergraduate, came as a surprise to me because electricity was something that I thought of as being dangerous and a little bit scary. But in fact, our brains and our bodies use electricity all of the time. And it's not just our bodies, but luckily for those of us that want to study this, lab mice also use electricity, as do horses and penguins and sharks, and basically, I'm just pulling up good and funny examples. Every other animal you can think of uses electricity in their body. And indeed, it's not just animals, but the bluebonnets and the cacti and the oak trees that you see around us, all of the plants on the earth use electricity all the time, and mushrooms and mold and fungi, and protozoans and bacteria. And basically what I'm getting at is every living cell makes use of electricity, and does so in a really interesting way. So this is where we're going to start tonight, and we're going to work from this level of electricity and how living cells use electricity all the way up to epilepsy, which is itself a set of disorders that are directly linked to how our bodies and how our brains use electricity. So our brain is very specifically an electric organ. It makes use of electricity in a way that's really refined above and beyond the other cells of our body. And we know that our muscles make use of electricity as well, but our brains are really known as kind of the most electrical of organs. And so we're going to talk a lot about how that works. And that this electricity is vital to the function of our brains and to brain function. And since I hear just a little bit of feedback really briefly, I wanted to mention that the audiovisual people warned me that sometimes cell phones cause feedback through our wireless microphones. So putting cell phones on silent mode, or on airplane mode, if you don't mind, can sometimes cut down on it a little bit. So if you get a chance, you can put your phones on airplane mode. It will keep our communication a little freer here. But electricity is vital to the function of the cells in our brain. And to really understand this, we have to move beyond just the brain itself and delve down deeper into the smaller and smaller structures that make up our brain. So our brain is this organ that sits inside of our skull, and it's a really beautiful organ. It's made up of all of these different types of tissues and different types of cells in different areas. And we can make really beautiful pictures of it. And as we zoom in closer and closer, what we see is that the individual cells of our brain connect together in very specific ways, and very interesting ways. And so we've moved down from the brain to an expanded view of the brain and to a group of these cells, all clumped together and shown in different colors, and then up to a single cell. And this is a single brain cell. And every point of yellow along the surface of this cell, as it branches out, these finger-like projections. Every one of these yellow spots is what's called a synapse. And that's a connection between this cell and one of its neighbors, or a cell in a different part of the brain. And it's the connection between those synapses that allows neural communication to occur, that allows us to think, and allows us to perceive our environment, and allows us to store and retain our memories. It's through those connections and the electrical activity of those connections that thought and cognition can occur. And so this is what we're going to talk about tonight. And so we're going to approach this in 3 different chapters, because in order to talk about electricity and how our brain uses it, I want to make sure we're on the same page. So we're going to start off talking about electricity itself and bioelectricity and how it is that a cell in our brain can make use of electricity as a way to send signals. Then we're going to move on and talk specifically about electrical brain signaling, and then we'll talk about the larger disruptions in brain signaling. And I'll focus mostly on epilepsy, which is a set of disorders that I specifically study in my lab, and the electrical brainstorms that epilepsy induces in our brains, and how these things are all linked together. So to start, to bring us down to basic principles, if there are any engineers in the audience, they might roll their eyes a little bit at this, but that's fine. We have to start at the level of atoms. So atoms are made up of different particles with different charges. So the main ones we need to focus on are the electrons, which circle around the outside and have a negative charge. And they circle around the nucleus, which is made up of protons with positive charges and neutrons that have no charge. And so there are these different particles that make up the atoms, that make up everything around us. So what electricity is really just the movement of some of these small particles. So these electrons, they carry a negative charge. If you push them with the right amount of force, you can bounce them around. And that negative charge, that particle, will bounce from one atom over to the next. And so you can make that charged particle move. And if you can string together a bunch of these movements and make that electron jump and jump and jump, we have charged particles moving and flowing. And that is electricity. That's all electricity is you just need charged particles to move and to flow in a specific direction. And you've got electricity. So it's as simple as that. And this is something that in a similar way our body can take advantage of. And as you think about everything that plugs into the wall, can take advantage of. So just to throw some jargon out there, some words that you might recognize or that might sound familiar, this movement of electrons is called the flow of electricity, or the flow of electrical current. And you can think of it like a river of these electrons flowing along. So how do they flow? And how do we contain and make use of that flow? So there are different types of materials that allow that flow or don't allow it in different ways. So we can start off with a conductor. So this is a material that allows electricity to flow easily. So you can think of like copper or metals. And the opposite of that would be a resistor. A resistor is a material through which electricity flows poorly. And if you take a conductor like copper wire and you coat it with a resistor like plastic, that's what you've got. You've got a wire and you've got this nice pathway down which electricity can flow. So I want you to keep that in mind as we go and we start talking about cells and things that are much more biological than a piece of copper wiring. A wire is really just something that allows electricity to flow. It's an easy path that those charged particles can move on, surrounded by a resistor that protects and directs that flow of charged particles. And that's electricity, and that's something that our cells can take advantage of. We'll talk about that in a minute. So the other major kind of basic principle I want to talk about is storing electricity. And we store electricity really easily in a battery. So a battery, as you all know, I've used one and plugged one in, has a negative side and a positive side. And this is labeled on purpose to line up with those charged particles that we saw when we looked at an atom. Because this negative side is just made up of material that can give up electrons. Whereas this positive side is made up of material that can absorb electrons. And this is going to encourage the flow of electricity from one side to the other. So a battery stores electricity so that it can be used later for something else. We just have to plug it in and attach it to some wires. And this all will become clear as to how this relates to our biology in just a minute. But electrons are tricky to hold on to. And they're small and they move extremely quickly. They're hard to control. But our body takes advantage of a different type of charged particle in order to use electricity for signaling. And instead of using electrons, our body actually takes advantage of something that surrounds us and is present in our environment and that our cells are really easy or are really good at taking advantage of. And that is salts. So salts are a really interesting type of substance. You can think of the most standard salt is the stuff that sits on your table and you sprinkle on your food, which is sodium chloride. Sodium chloride is table salt. It's made up of two things, hence the name, sodium and chloride. So sodium chloride does an interesting thing, and salts do an interesting thing when you put them into water. If you put sugar into water, it dissolves, disappears, and you can't see it anymore. But each of those molecules is still there, but each of those molecules is still sugar. So you have sugar dissolved in water, you have a bunch of little tiny sugars broken down into the components. Salt does something different when you put it into water. And that's that it breaks apart. Instead of staying together as sodium chloride, when you put salt into water, it dissolves into its individual ions. So now you have water that's full of sodium and it's full of chloride. And those things aren't really interacting in the same way anymore. They've dissolved and they've separated into ions, and you can see that I've added something up here which matches up with the symbols that we showed with protons and electrons and that we showed with the battery, which are this plus sign and this minus sign. Because these are charged particles, just like a proton and electron. A sodium is missing an electron, so it's positively charged, and this chloride has an extra one, has an extra electron, so it's negatively charged. And our cells are really good at dealing with salts because they're surrounded with salts all the time. So again, this is our sodium. A positive charged ion is missing some electrons, whereas this negative ion has an extra one. But just like when an electron moves down a wire, bouncing along and flowing, becoming electricity, the same thing can happen when you move a different type of charged particle, one of these ions. So if we move this sodium ion around in the water, that is electricity. That is the movement of a charged particle in the same way that an electron flowing down a wire is electricity. And this is something that our cells are really good at taking advantage of. They aren't good at controlling electrons, but our cells are really good at controlling these bigger ions, these salts. So, how do they do this? This is kind of the mystery about how cells can use electricity as a form of signaling. Is that they actually, instead of using electrons, they use these salts as their source of electricity, and they're very good at controlling them. And they can do this because cells are surrounded by salty water. So what is our blood that pumps through our body and surrounds all of our cells? It's water with red blood cells and salts, and probably a little bit of sugar, hopefully not too much, mixed in there. Inside of our cells is more salty water, a different kind of salty water. So how is it that cells can then use these ions to harness electricity to send signals? So this is what we're going to talk about next. So we're going to move from this really complicated looking cell, which is really beautiful, but it's hard to keep making figures for. So I'm just going to use kind of this cartoony looking cell. This is my little cartoon of brain cell. And it's sitting inside of our brain, and it's surrounded by this salty water. That salty water means it's got lots of ions. It's got lots of sodium and lots of chloride. It's got other things as well, like potassium, other types of salts. But it's constantly just sitting in it, soaking in it. And then inside that cell is, like I said, another set of salty water and lots more ions. But the trick about the cell is that there's something in between that outside and that inside. And that's the cell membrane. And the cell membrane is what contains everything within the cell, keeps everything packaged together. And that cell membrane is a fantastic resistor. So now we have the situation, like I described before, where we have a good conductor, which is salt water, and encased within a resistor. And if this sounds familiar from a couple of slides ago, that means that the cell is really good at behaving like a wire. It's very good at conducting electrical signals because it has a conductor on the inside encased in a resistor, just like a wire. And so this is the first way that these cells are able to behave electrically, because they contain a source of electricity, those ions, and they're able to pass it and move it around because the inside of the cells are really good at moving those ions. So how does this do this? The cell actually has a type of machinery to transport the ions and to move them around where it wants. So in this example, it can grasp onto a certain type of ion, and then it can transport that ion to the inside of the cell. And now it has moved that charged particle. And movement of charged particles is electricity. But in addition to moving that charged particle, now it has separated these two charged particles, and it's storing them. And what is it that stores electricity but a battery? So the cell can behave both as a wire to transmit and allow electricity to flow, but it can also behave as a battery to store that electricity and use it whenever it wants. And this is the key to electrical signaling by cells. So the cell uses ions, which are just salts dissolved in the water, in the plasma that surrounds them, as its form of bioelectricity. And it can store them like a battery. And because it is such a good conductor surrounded by a strong resistor, it can behave like a wire to transmit those electrical signals whenever it wants. So the question now is, a cell can store electricity and it can transmit it, but what does it use that bioelectricity for? And what is the purpose of electrical signaling by the cells? So this brings us to the second chapter of our talk, which is actual electrical brain signaling and what the cell does with these electrical signals. All right, so the main thing that these cells do, these cells in our brain, with these electrical signals, is they send signals between each other. And they transmit signals from one cell to the next. So once one cell activates, it can send one of these electrical signals, powered by its own cellular battery, down those projecting fingers that you saw in that image, to the next cell down the line. So it's really good at then transmitting the signal using that electrical charge that it maintains with its cellular battery. And at that connection between the cells, and I describe those connections as synapses, it goes through a really interesting process. When that electrical signal comes, to this part of the synapse, it signals to the cell to release a chemical. And this chemical is called a neurotransmitter. And the neurotransmitter is ejected from the cell, and it's sensed by the next cell in the chain. And those neurotransmitters activate the battery of the next cell down the chain and cause it to send its own electrical signal. So in this sense, what a synapse is doing is taking an electrical signal, turning it into a chemical signal, which the next cell in the line then reverts back to an electrical signal. And this is the process that we call synaptic transmission. It's taking the signal in one cell, a cell is activated and sending an electrical pulse, turning it into this chemical neurotransmitter signal, which the next cell detects and turns back into an electrical signal. And the processing that our brains do at the level of cells is each individual cell is receiving tens of thousands of these signals at any given time. And this cell needs to interpret these signals and make a decision as to when it has received enough signals that it wants to then send its own signal. And this is neural processing, is how cells combine all of their signals together and make that decision to then transmit that signal down the chain and down the path. And this is how we perceive our environment, through our visual system and our auditory system. This is how we consolidate and recall memories. This is how we do everything that our brain does, is through these processes of electrical signaling by all of these cells. So why do we use, or why do the cells use electricity for this type of signaling? If we've got these chemical neurotransmitters, why don't the cells just use that entirely? And there's an interesting answer to this. So what we have is this connection between cells, this synapse, is an electrical signal turning into a chemical signal and turning back into an electrical signal. But this chemical signal needs to diffuse between one cell and the next because these cells aren't actually touching together. There's a gap between them across the synapse. And diffusion is really great for these small distances across the synapse. So I just put this little video up here to show you an example of diffusion. Diffusion is great across a small synapse, but as you can see, if we're just putting dye into a glass, diffusion is a really inefficient way to signal across big distances. So the synapse itself is 0.0000003 meters across. Diffusion is really, really efficient to send that signal from one cell to the next. But our bodies are much larger than that. And diffusion, as you can see just from this example in a glass of water, it takes much too long for a signal to diffuse throughout that entire glass of water before a signal would arrive anywhere. And the things that our brains are doing and the control that's required for our bodies requires a signal that happens much faster than this diffusion. So diffusion isn't fast enough for movement and sensory perception and cognition. And if we want to do complex movements and complex cognition, like is shown up here, you need a fast signal. Diffusion isn't adequate. And this is why an electrical signal is an amazing way for our cells to send these types of processes. Because we want to control our bodies and we want to control our brains at a much faster level than diffusion can do. So neural signals have to cover a lot of distance. We have nerve fibers in our toes that stretch all the way up to our spinal cord, which then stretch all the way up to our brain. That's a lot of distance to cover, and diffusion isn't good enough for that. So by using electrical signals, we can overcome that slow process of diffusion and get the precise timing that's required for us to stand and to dance, and the precise timing that's required for us to understand speech or decode a visual scene. So only bioelectrical signals are really fast enough for that neural processing. So neural processing is very complicated, and what I've shown you are much more, very simplified versions of this. We draw these little cartoony figures because that's nice to explain how a single synapse works. But if you recall from this actual picture of a cell, there are, even in this little image, hundreds of these yellow spots, hundreds of these synapses. And many cells in our body contain tens of thousands of synapses, tens of thousands of connections with each other, all of which are sending signals, all of which are processing those signals and listening to those signals and trying to understand and collect information out of them. So the realistic situation is, that my slides advance without me pressing the button, is that neural processing is a really complicated thing. And when we zoom out and stop looking at individual cells, we start to see how complicated this process really is. So when we move from single cells and think about how cells actually process signals in the brain, we move to what are called neural circuits. Neural circuits are groups of cells that are linked together by these synapses. Cells that do the same job, that do the same type of processing. And so now we'll talk a little bit about how those neural circuits work in this process. So different cells in the brain that are connected in a neural circuit work together. And they work together to accomplish a job, to do the same thing. So if we start with a signal in one cell, and as you can see, I'm no graphic designer. After I made these slides, I realized that these looked kind of like psychedelic race turtles. But these are individual cells. And if we start with a signal here, an electrical signal in this cell, it's going to send that signal throughout the neural circuit. And that neural circuit might encode a memory, or it might encode speech, allow us to understand language, or allow us to understand visual signals that we're receiving. But these cells working together form a circuit. And when you get a bunch of cells working together, all of which are electrically active, you can actually detect that electrical activity from outside the brain. And we do that with something that's called an EEG. So an EEG, which is an electroencephalogram, is something that might be familiar to some of you, but it might be more familiar to hear about an EKG, which is an electrocardiogram, which is detecting the electrical signals from our hearts. So this is probably familiar to everyone from movies and TV, and maybe if we're unlucky from some personal experiences. But this is a recording of the electrical activity of our heart. Because like I said, muscles, which our heart is a muscle, uses electricity just the way, or in a very similar way that our brain does. But just as we can record this EKG by sticking electrodes on our skin in different parts of our body, we can record electrical activity of our brains by sticking electrodes on our scalp. And we get something that looks like this. And so these are the brain waves recorded at each of these different points across the scalp. And it's really amazing that we can record these things through our thick skulls and our skin and everything that is between those electrodes that are stuck to our skin and our brain itself. But each wiggle in this EEG signal is cells sending electrical signals. So in order to make this EEG wiggle, as you can imagine, these cells are all very tiny. It takes thousands of cells acting together just to evoke this activity that you see in the EEG trace. And so when you see these bigger waves, this is a whole lot of cells in your brain acting together and acting synchronously. And the smaller wiggles are when the cells are active, but they're active asynchronously. They're not active at the same time. And just by looking at the waves in these traces, we can infer some things about the mental or conscious state of our brain. So when we see a lot of this fast, spiky activity, which we would call high frequency activity, this is the sign that our brain is awake and it's thinking. And what this usually means is that the neural circuits are highly active, but they're active in such a way that they're processing a lot of things, which tends to mean that they're doing it asynchronously. They're desynchronized in order to do that complex processing. As our kind of level of consciousness goes downward from awake and thinking hard to awake and resting, to drowsy and sleep and deep sleep, you see that these brain waves change. They change their shape. They tend to get bigger and they tend to get slower. And this just indicates that the neural circuits are kind of relaxing and they're starting to just behave synchronously. And they're oscillating back and forth in a much slower way. And so we can infer what our brain is actually doing based on just the shape and the size of the waves that we see in the electroencephalogram, the EEG traces. But the EEG traces can tell us other things as well. And that's what we're going to talk about in the next section of this, is what happens when we see abnormal EEG activity. When our EEG traces come along in a normal-looking rhythm, and then they break down and they show us something quite different. And this is where we get to the part of the talk where we talk about epilepsy and other neurological and psychiatric diseases that show abnormalities on the EEG. And the reason that you can detect these abnormalities on the EEG is because they're related to a dysfunction of electrical activities in our brains. So epilepsy, or electrical brainstorms, and why epilepsy happens and how it happens is kind of the next section of this talk. So, I normally don't like to put up too many slides that just show facts and figures, but I think that this is really important to go through what epilepsy is and what it is that we're talking about and how prevalent this is before we talk about any of the causes. So, what epilepsy actually is, or you can receive a diagnosis of epilepsy just by virtue of having more than one seizure within a year that is unprovoked and not caused by some acute medical condition. So your body can seize. You can undergo a seizure due to low blood sugar or due to withdrawal from drugs or alcohol, but that isn't the type of seizure we're talking about. We're talking about a seizure that is not directly linked to an acute medical condition and isn't something that's directly provoked. And if you have more than one of those, separated by more than 24 hours, but within a year, you have epilepsy. So you can imagine just from that fact that epilepsy is a really broad spectrum of conditions. It can be someone who in adulthood experiences one seizure and then 6 months later experiences another seizure. It can be an infant who experiences many seizures during the course of a day. And so these epilepsies are very different disorders. They're very different types of conditions. But they all fall under this umbrella of epilepsy. They're all disorders that are linked to having seizures. So this is a very common condition as well. So this is the fourth most common neurological condition behind migraine and stroke, and another one that I don't study. That I think is Alzheimer's. But either way, it's a very common condition. And so common that 1 in 26 people throughout the world will be diagnosed with epilepsy at some point during their lifetime. But epilepsy, of course, it actually can resolve. So currently we estimate that there are 3.4 million people in the United States living actively with epilepsy. So it's an extremely common condition. It's an extremely costly condition. And for some people, it's a very manageable condition, and for others, it is much less. But related to that, only 1 in 3 people with epilepsy, or 1 in 3 people with epilepsy report, they don't feel that they have full medical control of their condition. And that can mean that the epileptic drugs that they're taking, the anti-epileptic drugs that they're taking, aren't effective at blocking their seizures. Or it could mean that they've taken anti-epileptic drugs and find that the side effects are too overwhelming, that they can't deal with taking those drugs, and they still aren't able to control their condition. So it's a very common condition, and it's something that we don't really have a good control of. So what causes epilepsy? As I said, it's a really broad spectrum of conditions, and it has a really broad spectrum of causes. So these can be genetic. It can be related to brain trauma or infection or stroke. There's a whole bunch of other causes that I just couldn't list them all. And it can be caused by developmental changes. But really importantly, up to 60% of the patients that come in to a clinic with epilepsy are of unknown cause. So we don't understand the etiology or the cause of the majority of epilepsy patients that come into the clinic. And a huge number of those patients we still have a hard time treating. So this is a really important disease that we struggle to understand. So what is a seizure? A seizure is kind of very simply, it's a change in electrical activity in the brain. And you can see we've kind of got this one in the background, that you can see that there's a fundamental change in this EEG waveform that we can actually pick up and record electrically. And this change in the electrical activity causes some change in how we feel or how we act. Epilepsy doesn't necessarily cause a loss of consciousness. It doesn't necessarily cause us to lose posture and fall to the ground or have uncontrolled body movements. But as a neurologist told me recently, which I thought was an interesting way of putting it, anything your brain can do, a seizure can do. So your brain controls sights, sounds, smells, and emotions, and movements. Any of those things, a seizure can take the form of those things. So you might have a seizure that doesn't cause the loss of consciousness, it just causes strange sensations. Or it just causes focal motor control difficulties. It could cause complete loss of consciousness. But all of these things can be seizures. So seizures themselves are a really broad range of different things. So we can't really define seizures as any one thing. All of these different types, they're all different types of seizures that are related to all these different types of disorders and all the different causes of epilepsy. But what we can start to understand is what it is that makes seizures happen. And this is where we go back from the brain and the circuits and the EEG, back down to the level of the cells. And we can look at the cells and start to understand what it is that's happening at the level of that electrical cellular signaling that's changing and causing these disorders. So we can start with different forms of altered cell signaling that we've discussed. There's certain types of changes to the cell's battery that can alter how how our brain cells function and can lead to seizures and epilepsy. There are changes to how the cell sends its electrical signals. They can lead to epilepsy. And there are changes to these chemical signals at the synapse that can lead to epilepsy. All of these parts in this important chain of signaling that a cell goes through are susceptible to dysfunction that can lead to epilepsy. One thing that I didn't discuss before is that within a neural circuit, there are signals that tell cells to go, and there are signals that tell cells to stop. And if we change the balance of those two things, we can also lead the brain down a path towards epilepsy. So if we have too much of a go signal, which we would call too much excitation, you can end up with epilepsy. And on the opposite side, if we have not enough of that stop signal within a neural circuit, we can end up with epilepsy. And finally, the connections between cells can be altered. So not just the signaling, but the connections themselves can be altered to cause epilepsy. So if we normally have a situation where one cell links to a bunch of partners throughout a circuit to perform a task, just very subtle changes in the links between these cells, just the addition of a few extra connections, can lead to the recruitment of a bunch of extra cells into this process. And this itself can lead to overactivation of certain brain circuits, and this can lead to epilepsy. And as you may or may not have heard, for many patients, epilepsy becomes a progressive disease, where the seizures actually get worse over time. And this can be because this activity that's driven by epilepsy can lead to further changes within the neural circuitry, the addition of new connections, which over time will make the neural circuit behave worse and worse and worse. So in these cases, epilepsy can actually progress and change over the course of a patient's lifetime. So what actually causes epilepsy? At kind of the body level, are these things that I mentioned before. We've got genetic causes, we've got stroke and brain trauma and infection, developmental causes, and a bunch of unknown causes. But what actually makes epilepsy happen occurs at the level of our cells. And that can be changes to the electrical signaling that is necessary for our brains to function. So it can be changes to a cell's battery, it can be changes to the electrical or chemical signaling of the cells, and changes to how the cells are connected together within the circuit. Oh, excuse me. So, how do we then treat epilepsy? If we know that some of these things happen at the level of the cells, some of the drugs that are effective at treating epilepsy actually target these very processes at the levels of the cells. So we have whole families of epilepsy drugs that are focused on changing the electrical signaling by individual cells. And I'm not going to expect you to know the names of some of these drugs, but there are multiple different types of drugs that can be given to patients that will change the electrical signaling in their brain cells in different ways in order to try to stop their epilepsy. There are drugs that affect this chemical signaling between cells, the synaptic transmission process that I described. And one of the things that you might notice is that while a lot of these drug names are unfamiliar and they just sound like most drug names do, there are some that might sound familiar, which are benzodiazepines or barbiturates. Because it's important to notice that these processes that these cells are involved with are not just involved in epilepsy, but these are the same processes that can be affected in other neurological diseases and other psychiatric diseases. So some of the drugs that are used to treat epilepsy can also be effective in treating other diseases as well. And finally, there are potentially drugs that can alter the cell's battery themselves. And I wanted to bring this up specifically because we just had a talk here at UT by someone that the Department of Neurology is hoping to recruit who does specific research on how cells handle chloride, as in sodium chloride and table salt. And that altering how cells handle chloride might be a key to treating certain types of really drug-resistant epilepsies. So, discovering new drugs requires understanding what it is that isn't working in our cells, and what about these processes are broken in order to understand how we can correct these things. So this is where research comes in. So I want to talk very quickly about the research that happens in my lab. My lab does not study epilepsy in people, but we study epilepsy in epileptic mice here in the Department of Neuroscience at UT. So the things that I look at in lab are the electrical signaling and the chemical signaling of individual brain cells in these epileptic mice. And these are genetically engineered mice to have the same types of mutations that affect certain pediatric epilepsy patients. So this is actually a recording made in my lab by my wonderful postdoc, Jessica Chansey. And what you can see here, it's a little bit dim, but this is the main cell body here that would be shown right here. And these are these long finger-like projections that a lot of brain cells have that are called dendrites. This big triangular thing is actually one of our field's most high-tech voltage sensors. And we can actually put this voltage sensor down on an individual cell and record the electrical signals that that cell is making and try to understand what has changed in these epileptic cells and what we might be able to attack with new types of drugs to change the electrical signaling back to a more normal type. So each of these spikes in these black traces is an electrical signal that this cell is sending. And it becomes harder to see, but there are actually small bumps in this trace, and these little bumps that you can see, especially down here, are the signals of individual synapses firing away as we record from this cell. This is a live cell recorded in the brain of a mouse. And we can record individual synapses, sending their chemical signals that are then being reverted back into electrical signals by the cell that we're monitoring. So the goal of the work in my lab is to understand how these epilepsy genes can change the electrical signaling and the chemical signaling and then the neural processing within the brain circuits of these mice in order to understand the processes that we might attack in the future and discover new drugs that can help patients with these types of epilepsy. So the people that work in my lab are Jessica Chansey, who is my postdoc, who does really amazing electrical recordings from cells from epileptic mice. Aurora and Meredith and Simon and Alexandra are newcomers to the lab. We're learning these techniques. And then I wanted to mention my collaborators in Audrey Brumbach's lab, who actually gave one of these brainstorm talks earlier in the year during the fall. And her work is on autism. And my lab is in the Department for Neuroscience, or the Department of Neuroscience, and then I also have an appointment in neurology at the Dell Medical School and in the Center for Learning and Memory. And most of all, I want to thank you guys for coming out and listening to this talk, and we really welcome any type of question that you might have for us. We'll take a 5 to 10 minute break, and then let you know when it's time to come back in, and we'd love to hear your questions and any questions you have for the panel. So thank you very much.